Hi, my name is Veronika Müller, and I will start with a general introduction into neuroimaging meta-analysis, examples of applications, and in particular show some studies in major depression. Over the last 2 decades, neuroimaging has evolved into a widely used method to investigate brain structure and function, as well as the pathophysiology of brain disorders. Since the beginning of neuroimaging, the number of studies has steadily increased, with now more than 3,000 published fMRI studies every year. However, in the last few years, it has more and more been shown that a lot of published effects are not replicable. For structural brain behavior associations, for example, only little spatial overlap has been found across results of independent samples. Similarly, for task fMRI data, low overlap of thresholded statistical maps of the same contrasts between different independent samples have been reported. All of these studies showed that this lack of overlap decreases with increasing sample size. In general, single task-based studies, especially those testing for altered brain activation in clinical samples, often rely on small sample sizes. This may on the one hand lead to missed results, but on the other hand also push researchers to analysis that increase false positive findings. Furthermore, a lack of replicability might be due to heterogeneity between studies, as they use a wide range of different designs, procedures, and analysis to assess the same mental process. Interference processing, for example, is investigated by using the original color word interference test, but there are also a number of other versions of the tasks. Additionally, studies also vary in their analysis approaches, like pre-processing pipelines, software packages, and statistics. A large study in 2020 demonstrated that analysis of the same data and hypotheses by 70 independent teams revealed high levels of disagreement. Therefore, insights from individual neuroimaging studies are limited by study-specific effects and characteristics due to low sample size and flexibility in analysis and experimental designs, but also selective reporting and p-hacking should be mentioned here. This makes it increasingly challenging to keep track of the wealth of findings and to distinguish spurious from replicable findings. Therefore, there is the need to quantitatively consolidate effects across individual studies to assess what has been consistently found. Until now, I have only showed the problems of individual studies. However, there are many neuroimaging studies, and despite the analytical flexibility, one of the things that they all have in common is the use of a standardized stereotactic space, reporting results as X, Y, Z coordinates in MNI or Talara space. And here meta-analyses come into play. They synthesize the multitude of results in standardized space and assess the brain regions consistently reported across experiments. There are 2 general approaches to neuroimaging meta-analysis. Image-based analysis combine raw data or the full statistical images of the original studies, which has the drawback that it is often hard to compile a dataset that is representative of the literature. In contrast, coordinate-based meta-analyses are based on published coordinates of activation peaks, with the drawback of a sparser representation of the findings. However, as most publications report their findings as coordinates, coordinate-based meta-analysis allow to capitalize on most of the published neuroimaging literature on a specific topic. In brief, coordinate-based meta-analysis extract from each publication that investigates the topic of interest all the reported coordinates. Those coordinates are then taken, the spatial uncertainty modeled based on between subject and template variance, and the voxel-wise probabilities combined across experiments. These are then tested against the null distribution of random spatial association between experiments, thresholded and corrected for multiple comparisons. Results therefore reflect regions that are consistently found across experiments. This makes it possible to identify robust networks involved in a specific mental task or process. We have, for example, derived a network consistently shown to be activated across neuroimaging studies of face processing, different facets of empathy, or during impulsive behavior. Beyond robust network associated to a specific mental process, differences in brain activation of brain structure between groups, for example, a clinical population compared to healthy controls, can also be tested for spatial consistency across studies. The lab of Atkin did quite some work there, consolidating most of the psychiatric literature and showing in a transdiagnostic approach that there's robust convergence across different psychiatric diseases in meta alterations, but also task activation alterations. We identified regions consistently reported to be associated to formal thought disorder, and then used those regions as seeds for functional connectivity analysis to compare patients with schizophrenia and healthy controls. And this is another interesting field of application of meta-analysis, to use regions that are consistently involved in specific processes or group alterations as an informed network for further investigation. What makes this approach so attractive is a direct link to a specific function, which often makes interpretation much easier. For example, we used a lot of different meta-analytically derived networks for classification of patients or prediction of clinical symptoms based on resting state functional connectivity within those networks. In a first study, we classified patients with schizophrenia, here shown in green, as well as Parkinson patients, shown in blue, from healthy comparison subjects, and found that different networks were differently informative for classifying Parkinson and schizophrenia, respectively. Additionally, individual symptoms in schizophrenia could be predicted by functional connectivity within meta-analytically derived network. In particular, cognitive symptoms could be predicted by connectivity within socio-affective networks. The remaining time of this talk, I want to focus on major depression and show different examples of the use and application of meta-analysis to gain insight into the neural correlates of this disease. In the first meta-analysis, we wanted to identify brain regions that are consistently reported as showing alterations in brain activation during emotional and cognitive tasks. However, surprisingly, we did not find any convergence at all when consolidating the findings across almost 100 experiments, neither in the analysis across all experiments, nor in specific sub-analysis, where we tried to analyze across more homogeneous types of tasks and samples. When looking at all those coordinates reported, we clearly can see that the findings are scattered all over the brain. So what can we learn from this not converging result? Importantly, the study in major depression is not the only meta-analysis with no significant effects. Inconsistent findings have, for example, also been reported for insomnia disorder or ADHD. There are many factors that contribute to these results, like uncorrected inference or publication biases, but especially heterogeneity of tasks, stimuli, designs, and analysis approaches plays a crucial role. With group comparisons, an additional layer of heterogeneity is introduced, and differences in investigated populations, medication, and comorbidity effects might play a stronger role than previously thought. Therefore, non-convergence in clinical meta-analysis can, on the one hand, result from problems and inconsistencies in the original literature, pointing to the need for more replication studies. On the other hand, it can also demonstrate the clinical picture of a disease, indicating that multiple different alterations go along with a particular disease with no clear or general pattern of change. In another meta-analysis in depression, we tried to reduce the heterogeneity by focusing on studies that didn't use a task and therefore only included VBM and resting state studies. We then found consistent changes in patients, in particular in the amygdala, hippocampus, and subgenual cingulate cortex. A recent meta-analysis consolidated the findings of intervention effects in depression across different modalities and therapies, and revealed consistent effects in the insular, amygdala, thalamus, and frontal regions. Importantly, results were strongly driven by resting and VBM studies, which again indicates that findings of task fMRI studies might induce a lot of heterogeneity. For dealing with the heterogeneity in task-based fMRI, we then went for a network-based approach. With meta-analysis, we had tested where in the brain localized activation changes in depression converge. We then thought that all those sites of aberrant processing in depression might converge in being connected to similar regions. That is, even though the clinical picture of aberrant brain sites is quite heterogeneous, it could be that those sites interact with the same regions. We did that by using the coordinates of the task-based meta-analysis in major depression, calculated resting state functional connectivity of each coordinate in 100 subjects of the Human Connectome project, and identified regions connected to those coordinates. The resulting networks therefore reflect the amount of integrity with sites of aberrant brain activation in depression. Interestingly, we found networks involved in emotion and cognitive processing, as well as previously been implicated in major depression. For example, highest integrities have been found for dorsolateral prefrontal cortex and cingulate regions, including subgenual cingulate cortex. Furthermore, integration of aberrant brain sites during task activation was also found with regions that were found to converge in the meta-analysis across structural and rest findings and treatment effects like cingulate cortex, hippocampus, and insula. We then compared those networks to previous networks models of depression by computing spatial correlations and found close similarities with our circuit. Therefore, with our meta-analysis, we could not localize any regions consistently found to show aberrant activation in major depression. However, what we see here is that quite a lot of those regions are connected to a specific network. Targeting this network therefore potentially affects all those regions dysregulated. This might explain why, for example, dorsolateral prefrontal cortex stimulation works well in depression, even though it is often not found to be specifically dysregulated. So to sum up this talk, meta-analyses are an important tool to delineate robust networks associated with a certain mental process or a disease. These can then be used as informed networks for further analysis, for example, in clinical populations. The lack of convergence allows conclusions about heterogeneity of individual studies or provides information on the clinical picture of a disease. However, either way, the fact that especially in clinical studies, meta-analyses often do not converge also points to the need for more replication studies, pre-registrations, and to follow best practice guidelines. From consolidation of the neuroimaging literature in major depression, we can derive that depression is associated to changes in regions of affective and cognitive processing, especially the amygdala, subgenual cingulate cortex, and hippocampus, and that some of those changes, like for example the amygdala, are consistently found to be affected by treatment. However, for alterations in task activation in depression, there are no regions consistently found across studies. But when looking at those findings from a network perspective, we found a network connected to the sites of inconsistent task activations, alterations in depression. And with that, I want to especially thank Simon Eickhoff, my group, and people of our institute in Jülich and Düsseldorf, all collaborators, and you for your attention. And if you're interested in this topic, come and see these posters presented here at OHBM.